All right, so this lecture video is going to cover specifically Mendel's third principle, okay, that's called the principle of independent assortment. And he figured this one out when he studied the possible combinations in his pea plants when he was looking at 2 traits at one time, 3 traits at one time. Okay, that's how he figured out this particular principle. So this is called the principle of independent assortment. And what this means is that the genes for different traits are going to segregate independently of each other during the formation of gametes, all right? And this is what's going to yield to the vast genetic diversity we have in sexually reproducing species, all right? So your example here, it says the inheritance of pea color is completely independent of the inheritance of the height of Mendel's plants, all right? So what does that mean? Okay, well, for us to do this in class, we're going to have to use what's called a dihybrid cross. So when we looked at one trait, we were using a monohybrid cross where our Punnett squares had 4 boxes. Now to study 2 traits at one time, the Punnett square is going to have 16 possible combinations in the offspring, right? So we're going to work with this particular example here. It's pretty easy to see what's going on. So we've got Mendel's pea plants where he was going to study whether the peas were round or wrinkled. Okay, that's what the R's are for. And then whether they are yellow or green. Okay, so we're going to do, sorry, I'm going to scoot this out of the way for just a second. So we're going to do the capital R is going to be round. The lowercase r is going to be wrinkled. Capital Y is going to be yellow. Whoops, my W. And then a lowercase y is going to be green. Okay. So that being said, how in the world do we start? Okay, so notice that for each of these parents in this particular example, they're both going to be heterozygous for both traits, right? So to keep this simple, how do you take a genotype in the parents, whatever it happens to be, and figure out the alleles in the offspring, okay? So it's not going to matter which one I use here, all right, but we are going to write this here, and we're going to equate this to the FOIL concept in math, okay? FOIL stands for, if you have not covered this yet, first, outer, inner, last, okay, in math. So I'm going to work a math problem first, and I'm not going to get too worked up over or this might be inside and outside. Okay, so let's say that in math, your teacher tells you to simplify this, x plus 1 times x plus 3, okay? You're going to multiply your first, okay, which is going to give you x squared. You're going to then multiply your outer, your 3 times your x, okay, to simplify this, factor it. And you're going to multiply your inner, the 2 times the x, and then the last 2 times 3 gives you 6. Okay, now in math you would simplify this and you would get x squared plus 5x plus 6. Okay, we're not going to do that, right? We're going to say first, outer, inner, last, and leave it that way. Okay, so then we're going to do our genetics example. So over here we're going to put our r's in parentheses and our y's in parentheses. And we're going to do the same thing. Okay, so our first is going to be our big R and our big Y. Our outer is going to be our big R and our lowercase y. Okay, our inner is going to be our little r and our big Y. And our last is going to be lowercase r and lowercase y. Okay, now, a couple things here. These are not genotypes, right? These are alleles for 2 different traits in the gametes of the parents. Okay, so there's nothing that you can say about a genotype here. Right, you've got a dominant allele for each, a dominant and a recessive and a dominant, and a recessive and a recessive. Okay, so then in those particular parents, right, because they are the same, we can see, let me see if I can get, okay, so here's what I just wrote down following FOIL. There's my first, my outer, my inner, and my last. Okay, so I'm going to give you another example down here real quick. Let's say that we had this genotype. Okay, we put parentheses or imagine that they're there. My first is going to be that. My outer is going to be the same thing. So is my inner, this big R and that little Y. And then my last, sorry, bell's ringing, it's going to be this big R and this little Y. So that every possible gamete, whether it's egg or sperm, is going to only pass down these two alleles. Okay? So let's look back at our example up here. Okay, so then how do we fill this in? Right, so we've got our two R's, both dominant alleles, and our two Y's, both dominant alleles, where you're going to put your R's back together and put your Y's back together to make genotypes. So now we've got a homozygous dominant genotype for both of those traits. Okay, here, homozygous dominant genotype and a heterozygous genotype. Okay, so then we've got to figure out how in the world are we going to write a genotypic and a phenotypic ratio. Okay. And so if we look through this one, we end up, and this is kind of a hard one, I'm not going to write this out because it'll just take too long on a lecture video, but we only have one box that's this genotype, okay? So I'm just going to kind of walk you through the numbers here. You're going to have a 1, and I'm just going with genotypic ratio and just saying numbers, even though that's not how you would write it down. I'm going to have a 1 to 2, because those are the same, to 2 to 1 to 4 to 2 to 1 to 2 to 1 genotypic ratio. Okay, that would take you forever to write out. That's why I'm not doing it, right? That all those numbers add up to 16. Okay, you got to have a total of 16. You had to have a total of 4 in a monohybrid cross, right? Phenotypically, right, we're going to get all of these boxes are going to be yellow. Okay, and they're going to be round, right? In that little triangle. Let me see if I can write it here instead so we can still see it. So we're going to have 9 round yellow. And I'm just going to have to write them on top of each other. Okay, so then let's take care of all of our round offspring and get rid of them and not have to think about them anymore. So then we're going to have 3 round green. So we're done with round. Okay, then we're going to have 3 wrinkled yellow to 1 wrinkled green. Okay, so that's how you're going to work these, just kind of the quick example every time. You can also study 3 traits at a time, 4 traits at a time. When you get to 3, you use a mathematical formula, okay, instead of creating a Punnett square. And we don't do that in this particular class, okay? So when you think about this from a phenotypic standpoint, you have to get your essentially 4 possible combinations if they're present, okay? So you've got to get all of your round and yellow, and then all of your round and green in this example written down. Then you're done with the dominant phenotypic form, okay, of the shape. Then you take care of your recessive form of the shape. You get your recessive wrinkled and dominant yellow, okay, 3 of those, and then your both forms of the recessive gene phenotypically, right? So if you have any questions about dihybrid crosses, let me know, okay? Again, this is, well, let me say one more thing. This is based on the fact that every trait is going to be inherited independently of each other. Okay, the principle of segregation. The alleles segregate or separate independently of each other during meiosis. So let me know if you have any questions.